Hello, everyone. Welcome back to another CXC Cape Biology Past Paper Review. This is a Unit 1 paper. Actually, it's a Paper 3 paper, which is the alternative to your school-based assessment paper. So I've stumbled across this May-June 2013 paper, and I think there's some very useful information in this paper. So I'm going to run through it real quick, at least to try to help some of you in terms of some concepts that you might see popping up on your examination. As usual, my advice to you is always to read through the instructions carefully and make sure you understand all instructions before you start the examination. And if you have any queries or any doubt, you need to refer to your examiner to clarify all of those misconceptions or doubts before you start the examination. Again, this paper is for candidates who are doing the exam privately or unable to submit or do their SBAs. All right, and so let's jump into question number 1. And notice here they said, please begin question 1 first. And there's a reason for that. Based on how they set up their lab or the examination room, they want to actually do this first for a particular reason. All right. So also that you need to follow and make sure that you are conscious of all these instructions. All right. So anyhow, question number 1, part A1 is you're required to carry out a simple investigation determining the sucrose concentration, which is isotonic to potato tuber cells. It says you are provided with the following materials. So you have potato tuber cylinders. You have a 0.1, a 0.3, and a 0.6 molar concentration of sucrose solution. And you also have distilled water. Now here it said that the actual procedure here said here is a place the 0.1, the 0.3, the 0.6 molar concentration of sucrose solution and distilled water into 4 beakers labeled A, B, C, and D as shown in the table. And so looking at this table now carefully, you're going to realize now this is how you're actually going to label your stuff A to D, and then you label them according to your concentration right here. All right. So you don't have to put the concentration on the thing, you just label A, B, C, and D, and you know that these are the concentration of the sucrose solution. Now for part 2 now, or step 2, it said that remove any excess water from the potato cylinder using paper towel. Measure the initial length of the potato cylinders to the nearest centimeter and record the average length in table 1 and page 3. For the third step, it said place 3 cylinders into each. So notice now that there are 3 cylinders you're going to place in each of the beakers labeled A, B, C, and D and let stand for 1 hour. It said proceed to question 2 and 3. So notice now this is going to sit for 1 hour. And so that's why they wanted to move on to question 2 and 3 in the meantime. And so you come back and do the finish up the experiment. It's after 1 hour, remove the potato cylinder from the beakers and blot to remove excess solution. All right, so the 6th step now we said now measure the length of each potato cylinder in beaker A and calculate the average length. Record your answer in table 1 and page 3. Then it's a repeat step 6 for beakers B, C, and D. All right, again, you'll do all of these things here before we actually move on to question 2 and 3, right? And for each solution, calculate the average change in length and record your findings in table 1 and page 3. All right, so now let's go to the table. For this, I have something right here, but I'm gonna come back to that. I'm gonna mention that. But what I use here are some random numbers that I just think about. Matter of fact, these numbers are actually based on my experience, right? I was not in the experiment. I didn't know what was happening. So these are numbers that I use. I'm going to tell you why I use 5. So the initial length, or initial average length, I use 5 centimeters because typically to make this experiment pretty much accurate, you want to use anywhere between 4 to 5 centimeters. You can actually could use 6, not a problem. But I realized that when you go below 4 centimeters, it is kind of want to be less accurate because you have a shorter strip or a shorter cylinder, and so you tend to make more mistakes in terms of measurement, right? So you want to be a decent length that you can easily measure, right? Especially when you have a change in length. Now the final length, the 0.1, I expect that's supposed to be increased, and then for the 0.3, it will be pretty much the same, depending on the potato. And for 0.6, expect it to be a decrease in length. All right. And so in distilled water, that will be the greatest increase in length. Now, also, I want you to notice that for the decrease in length, I put a negative value there, right? And there's a reason why I do that, and it's for graphical purpose. When you're doing a graph, you'll be better to use a negative value on your graph, especially if you're looking at osmolarity. But I'm gonna talk about that as I get down into the question more. All right, so it says suggest a specific aim for your experiment, and for me, what I put here is that determine the concentration of sucrose solution in potato tuber cells, and pretty much that's what they want to do. Even though they gave you they actually gave you an aim, but based on the specific aim, what you want to do is to determine. You can say determine the osmolarity of potato tuber, or to determine the concentration of sucrose solution within that potato tuber itself, right? Now, matter of fact, they will accept any suitable or reasonable aim, So you don't have to be exact what I have, but anything that is reasonable and making sense, they will accept it. Now, it suggest an appropriate title for table 1. Write the answer on the lines provided above the table. So this is where this comes in now. And so this is a table. So a table showing the length and change in length of potato cylinders in different sugar concentrations. All right. So that's what the table actually showing. The difference in length and the length actually of the potatoes. All right. So here now for part 3, it's a account for the change in length of potato cylinders at 0.1 and 0.6 molarity of the solution. Now, at the 0.1, as I said, I expect there should be an increase in length, all right? When you're doing the experiment, actually doing the experiment, you can have something different. But, and again, this is also based up on the concentration within the potato itself. But based on experience, yeah, it will be an increase, or more likely an increase. All right, so here now, so the solution in the potato cylinder is more concentrated than the 0.1 molar of the sugar solution, right? So hence water moves into the potato cylinder, increasing the length of the potato. So that's what account for it. So water goes in. In fact, this solution will be called hypotonic solution. All right? And so which means that the solution in the beaker itself is more dilute compared to what is in the potato. So therefore, water moves from the higher water concentration to lower water concentration, which is from the beaker to the potato. Now, in the 0.6 molarity, the solution in potato cylinder is less concentrated than the 0.6 molarity of sugar solution. Hence, water moves out of the potato cylinder, decreasing the length of the potato. And in this case, this solution will be a hypertonic, which is PER. Okay, so it's hypertonic solution, which means that water moves from inside of a potato cylinder to the outside, where there is a higher concentration of the solute. All right, so let's go into the next part of the question now. And the next part of the question now reads now, describe 2 ways of using the data to estimate the osmolarity of the potato tuber. And notice here now, I actually talk about the negative value up there, which I'm going to express in the second option. Now, what you can do, you can use the concentration when there was no change in the potato length. That's very important. You can actually use to estimate it there. So this concentration is approximately, notice, approximately equal to the osmolarity of the potato. So since we classify where there's no change as isotonic, which means equal in concentration, what is inside the potato to what is outside of the potato, then at that point we can say the concentration in the potato equals to what is in the beaker. So it's an estimate. Again, it could be slightly different. Now, a more accurate way to do this is actually to plot a graph of the change in length. In some cases, if they give a mass, then mass will also be used, right? Because you can actually weigh the potato cylinders. But in this example or this experiment, they actually said measure. So what you're going to do is a graph again, length against the concentration of sucrose solution. And what you're going to draw is to draw a line of best fit, or what they call a trend line. Now, where the line intersects the x-axis, at that point, there's zero change in the y-axis, which indicating that the osmolarity, which indicate the value of the osmolarity of the potato. Or what you can also do based on this line of best fit or this trend line, you can use the formula y equal to mx plus b to solve for x. And x in this case will be the concentration of the isotonic solution or the osmolarity of the potato. All right. Again, this is more accurate than using the first option. Well, the first option is us to look and say, okay, yeah, this is where no change taking place. But again, it's not totally accurate because you can miss a point on your measurement, because it don't have to be exactly equal. It can be like a 0.00 something difference, right? So yeah, there will be a little bit of difference, but the second one is a little bit more accurate than the first. Again, isotonic point is also a key point to explain this, because again, everything is based upon the isotonic point, or when both concentration are equal. That's very important right there. All right. So next part of the question now is a figure 1 is an electron micrograph of organelles in a eukaryotic cell. All right, and so a eukaryotic cell is a complex cell, a cell like us that inside of us, multicellular organism. And so based on this micrograph, what this looks like is a nucleus. All right, so I can highlight something to you, and I'm going to point out something here. There's a reason why I highlight these two regions, right? They weren't highlighted, but I was highlighted for a reason. I'm going to take them off back for now. All right, okay. There's a reason why I highlight them. All right, and so what I want to point out here, X in particular, I like to be very specific. That's just me, right? And so they ask you to identify the structural label X and Y. And for me personally, X is pointing to the nuclear membrane of the nucleus. So for me to label this, what I'll say is the nuclear membrane of the nucleus. But they actually could look in for nucleus, right? But if it's a nuclear membrane of the nucleus, there's no way they could give it wrong, because it is exactly pointing to the nuclear membrane of the nucleus. Typically though, what most exams will show you, if they wanted to highlight the nucleus, is actually to label somewhere inside of here. So that's what I'm going to highlight here again. Anywhere here, if the line was here, then I say the nucleus. But nothing is wrong to say X is the nucleus, because it is actually the whole structure that X is on is actually the nucleus. This dark area right here is the nucleolus within the nucleus. So it's a mini nucleus right here that I'm highlighting right now. All right, so it's a mini nucleus type of thing. All right. Now, Y is closely attached or closely linked to X. And so if Y is very close to X, the only structure that could be, it is the rough endoplasmic reticulum, otherwise called the rough ER. All right? And that's the end of the first question. Now, for question number 2 now, it said, in an experiment, a pigeon pea plant with green round seeds and pigeon pea plant with white wrinkle seeds were crossed. It said all of the F1 plants had green round seeds. Now, all right, I'm going to talk about that. Let me just finish this part. It said the F1 plants were selfed, which means they are crossed with themselves. All right, so they are interbred. All right, so table 2 now is an incomplete table for the results for the F1 cross. So watch this now carefully. I want to point out something to you really quick. Now, if you have green and round seeds crossing with white and wrinkle seeds, right? Now, the only way you can get green and round here is that white and wrinkle must be recessive traits. Okay, they must be recessive traits. That means none of them are showing up in that cross, right? So they must be recessive. What you can also assume right here now is that the green and round, they must be, the green and round seeds must be homozygous dominant. Because if you have heterozygous green, then you're going to see some white showing up, right? If you have heterozygous round, then you're going to see some wrinkles showing up. So if you only see green and round, then we can now know that green and round, they are both, they are dominant to start with, but this genotype here must be homozygous dominant, all right? Now, so again, if we have homozygous dominant here and homozygous recessive, that we know for sure now that all the F1 progenies or offspring must be heterozygous. Because remember this, once you cross homozygous dominant with homozygous recessive, all the offspring must be heterozygous. So let's jump in the table now. I'm going to show you what I did here. Again, I kind of make a slight adjustment based on what the question actually asked me to do, but I want to point this out. I want you to pay attention to this because you really need to follow instruction in the examination. I kind of do it differently just to bring out a point, right? So here now, we're going to complete the table to show the expected phenotypic ratio for the cross. The phenotypic ratio, that's what we're going to show right here. Now, my thing is this. I'm going to show you the ratios. I put them on the side. This is actually the answer right here to be here. But I want to point out something to you. How I come by this. Now, the ratio in a dihybrid cross, once you have both parents, because remember now, all F1, they are heterozygous. And if you're going to cross two heterozygous together, as here right here said they are self, if you're going to cross two heterozygous as in dihybrid, the ratio is always a 9 to 3 to 3 to 1. You cannot change that. It will never change. Nothing can change it, right? So we guarantee you, I guarantee you, and you can check this out, once both parents are heterozygous in a dihybrid cross, the result, the phenotypic result, always be a 9 to 3 to 3 to 1. That can never change. That's standard. Now, so this is answer that's supposed to be in this column, okay? Now, the reason I put numbers in, I want to bring out another point. I really want to bring out this point because they could ask you anything, right? So, how do I get these numbers now? I look here and see there's a total right here, right? So if I get my ratio, I know my ratio, then I get my total right here. Let me go back to the last, to the phenotypic column with the numbers last. I'm going to show you something here. Now the total here is 1,136, right? That's the total number of offspring or progeny that you're going to get. Now my thing is this now. If this is equated to my 9 in my ratio, 780 is equated to my 9 in my ratio. My 3 is 148, and I'm going to tell you how I get this to be 3. And I know this is 3, I know this is 1, because again, they can mix up these things. For you to have both dominant traits, then you must have the highest number. That's a 9 ratio. Because green and round, they are dominant. We established that. Green is dominant, white is recessive. So once you have one dominant, one recessive, that's a 3. Again, white here is recessive. You know, they make a mistake right here. It can't be green and white, you know. They make a mistake right here. This is supposed to be green and wrinkle. Yeah, they make a mistake right here. They're supposed to be wrinkle. This is a typo. Let me just put that in. This is supposed to be wrinkle. Luckily, I see this. So this, they make a typo. It's a typo, so don't worry about it. Yeah, that's green and wrinkle. So you can't have two colors together, okay? So here now, green and wrinkle. So green is dominant, wrinkle is recessive. So once you have one dominant, one recessive, then you're going to have the 3 value of ratio. White and round. White again is recessive, round is the dominant. So again, once it's part recessive and part dominant, that's a 3. White and wrinkle, that's both recessive. So once you have both recessive together, that's the 1 ratio. So what I know for sure is that if my 9 ratio is 780 and my 3 ratio is 148, then my other 3 ratio must be 148. And then I add up all these numbers, and I add up all these numbers, which is 780, 148, and 148. I get 1,076. 1,076 minus from 1,136, I get 60. So I know that my 1 ratio will equate to a number of 60. Okay? Now, how do I get the values here? Again, I'm pointing out that the ratio, I put them on the side. The ratio is supposed to be here. But these numbers now, how do I get these numbers? I know my right ratio is 9 to 3 to 3 to 1, which are a total of 16. Okay? So what I did is divide the total by 16, as in we're doing a regular mathematics to find out the values of each ratio part. So you divide it by get 71. 71 times 9, you get 639. 71 times 3, you get 213. 71 times 3, 71 times 3, you get 213. And 71 times 1, you get 71. And so that's how I get these values and add up to 1,136 as well. So these are my expected numbers, because in some examination, they ask you to calculate the expected numbers. And I just want to point that out to you from using the ratio. Okay? So I hope this is clear and you can understand this. Now, for part 2 of this question, say, explain given 2 reasons why all F1 progenies were phenotypically alike. And one reason is that because the white and wrinkle are recessive traits, and white wrinkle seeds is homozygous recessive. The other part of the reason is that the green round parent seed, they were homozygous dominant, and hence all F1 offspring are heterozygous dominant. because the heterozygous dominant trait will overpower, I don't like using the word overpower, but will be prominent over the recessive trait, right? So they will suppress the recessive trait, in other words. All right. Or a big thing they can actually mention is that for all of the offspring, they will have the dominant allele. All right. And once the dominant allele are shown for both traits, then that's the only one that will show up. All right. So at least, so all of the offspring will have the dominant trait, because again, this is homozygous crossing with homozygous recessive. All right. So now part 3 here now. Is in the space provided below, construct a Punnett diagram for the test cross of F1 plant. I'm pointing a note right here that test cross means back cross, okay? And what back cross means is that you must use, back cross means that you must use a homozygous plant or parent to cross with the unknown that you're doing. So what we have here, we have the F1. We know at F1, they're all heterozygous. So it is given, okay? And so you're going to cross the F1 with the homozygous recessive. You're going to cross with homozygous recessive. And so here you say use the symbol G for color and R for shape of the seeds, including a diagram and indication of the genotypic ratios and phenotypic categories, for the phenotypic categories, as a matter of fact. So again, to do the test cross, you must use a homozygous recessive parent to cross it with whatever you're testing. All right? And so let's say they didn't know what F1 were, but we know that F1, based on what we got above, all F1, they are heterozygous. Now, I think what you're going to do here now is this. What I did is to break down the question a little bit further by saying that G is green because you know that green is dominant. Small g is white because you know that white is recessive. Uppercase R is round because you know that round was dominant. And small r for wrinkle because we know that wrinkle is recessive. Now, from the F1 plants, F1, they are all heterozygous. So therefore, this genotype here, which is big G, big R, small r, that is from F1. And from the homozygous recessive plant, now we're going to use to test the F1, must be homozygous recessive, which is small g, small r's. A point to note, from your heterozygous plant, what you will get as gametes will be uppercase G, uppercase R, uppercase G, uppercase R, lowercase r, I should say, lowercase G, uppercase R, lowercase G, lowercase r. How do I get this? Is by applying the law of segregation, or you can actually apply the law of independent assortment. Matter of fact, the law of independent assortment is a better one to actually apply here, matter of fact, because guess what? The colors, when they're forming gametes, the colors will not go together, is one off. In other words, that you have one of the pair of chromosome that will be separated from each genotype. All right? So what will happen here now? Again, G cannot go with G at all when they're forming gametes, period. Just remember that. So the uppercase G could go with uppercase R, the uppercase G could go with the lowercase r, the lowercase G could go with uppercase R, and the lowercase g could also go with the lowercase r. And that's why you have 4 different possible gametes for the F1 plants. Now, for the homozygous recessive, all the gametes must be small g, small r, because that's the only possibility you have. There's no other possibility. I only put 1, but you can't put 4 because there'll be 4 of them, okay? But I put 1. Now, the idea here now, I have the summarized version. You can use the expanded version if you want, but there's no need for that. That's really wasting of time. So the expanded version would have been, here you have 4 small g's and small r's. But I said there's one type. There's no need, right? So my Punnett diagram now will be here now. I have 4 columns and 1 row. If you want, you could have 4 rows, and each row would have small g, small r, right? All right. So what I did now, I put the gametes for 1, which is my F1 on top, and then at the row, I put my genotype, my gamete for my homozygous recessive. And so these are the results that I get right here, right? By doing the Punnett square, or complete the Punnett square. Now, based upon this, green and round will be 1 or 4. Why I put 4? Because if you do the expanded version, you'll get the 4. For my summarized version or reduced version, I get 1. Okay, one of each. All right, and for you, you get 4 of each. If you do the expanded version by putting the other 3 rows and put small g, small r's, again, there's no need to go through that. The ratios now will be 4 to 4 to 4 if you have the 16 box, or if you have the reduced version, it'll be 1 to 1 to 1. All right, because all the rows that follow this will also be the exact as the first row. All right, so that will be your ratio right there, and also your phenotype. A matter of fact, for this, your phenotype ratio and the genotype ratio will also be the same. They are both the same thing. All right, so here now for the next part question, so you're provided with a fresh slide preparation of onion root tip squash. Is in the box below, draw a cell undergoing metaphase of mitosis. And so what you know about metaphase of mitosis is that the chromosome, they will align in the middle of a cell called metaphase plate. All right, and they will align along that middle of a cell in single file. However, by viewing the slide, you may not catch division. You may not catch division at the exact midpoint of metaphase or the completion of metaphase. So what I did, I still put them in the middle of a cell, but I do not put them perfectly behind each other, because most times you're going to see them perfectly behind each other. But we know that they're going to be in single file, one after the other. That's the most important thing. All right, so you when looking at the slide, don't be alarmed or be confused if you see that some of the chromosomes are slightly away from each other. The idea is they're in one line in the middle of a cell along the metaphase plate in single file, one behind the other. That's the most important thing. All right? All right. And what I'm going to say to here, I know this is a 5-mark thing. What I'd wanted you to do when you're doing diagrams is please to label. So you're going to label the metaphase plate, you're going to label the cell, you're going to label the chromosomes, you're going to make mention to it, annotated diagrams. Please do not just draw a diagram and expect to get 5 marks. All right, so please annotate it. Draw your cell wall. So write the cell wall, write the metaphase plate, write the chromosomes, draw a diagram nice and neat. And actually, what you can also do is to give a magnification. They did not ask for magnification, but you can also give a magnification, right? There are 5 marks, so make sure you're in that 5 marks, right? Just don't put something on the page and say, okay, expect that 5 marks. Don't do that. Now, for last question here, it's a figure 2 is a photomicrograph of a reproductive cell. And so you can see the cell here. I label the parts here, but I'm going to go down because they show you a magnified version of it. And so the magnified version actually label again, which is the head, the mid piece here, and then we have the flagellum. All right, so here now it said that name the reproductive cell, and reproductive cell in this case is a sperm cell, or what they call a spermatozoon. For a singular, plural form will be protozoa, spermatozoa. Okay. Now identify the structure labeled A, B, and C, which I'll label those. So we have the head, we have the mid piece, we have the flagellum for 3 marks right there. For part 3 is a comment on the function of the structure labeled B. And the function of a structure labeled B, it provides energy for the cell to carry out its function, especially to swim and meet the ovum in the fallopian tube. So the general purpose here is really to provide energy or to produce energy. All right? And so the mid piece with the structure B, it contains a lot of mitochondria that will carry out respiration to provide needed energy or sufficient energy for the sperm cell to swim. Hence, in structure B, glucose and oxygen are converted, I can say react together to form ATP. Okay, so they react together to form ATP, which is a usable energy by the sperm cell. All right, so it's a metabolic process, which is a chemical reaction. All right. Now, part B, a table 3 shows data on the effectiveness of contraceptive methods. All right, methods expressed as percentage of women likely to become pregnant while using the method for 1 year. Now, if you notice the contraceptive method here, we have the male sterilization, which is vasectomy. We have the female sterilization, which is the tubal ligation. We have the pill, which is combined progesterone and estrogen. We have intrauterine device, which is IUD. We have the male condom, we have the female condom. We have spermicide. We have natural method, which include calendar, temperature, and the cervical mucus. All right, so let's go down now a little bit further now. So on the grid provided on page 9, construct a histogram for the data given in table 3. And now part 2 is determining the most effective and least effective method of conception. Cite the data used to justify your answer. And so for the most effective will be male sterilization, simply because it has the lowest percentage of pregnancy, which is 0.1%. For the least effective will be the spermicide, because the spermicide have the highest percentage of pregnancy, which is 18% based on the chart. All right. All right, so let's go to this. All right, so this is Instagram right here. So construct the Instagram, but I didn't show you the Instagram, so now I'm going to jump to the Instagram page right here now. So I want to point out something here now to get a marks for this as well. And this is what I really want to point out, is that what you need to do is label the graph properly. Matter of fact, use appropriate scales. All right, and the scales will make you have a nice large graph. The graph should not be too tiny that you cannot see properly. All right, or Instagram should not be small. You need to label the axis, all the axes, so the y-axis and the x-axis, you need to label both of them with proper units, and also proper units and title where necessary. So make sure you do that. And now here I put some abbreviation at the bottom right here on the x-axis. And so if you do this, you need to make a key to say, for example, MS. What MS is, all right, is which is male sterilization. FS, which is female sterilization. The pill, the IUDs, male condom, female condom, the spermicide, and the natural methods. So you need to actually put a key if you're going to put abbreviation. That's very important, right? All right, so let's go to the next part of the question now. It said, identify one method of conception from table 3 other than the male condom, which may give protection to HIV infection. Give one reason for your answer. And so the female condom is the only other one, right, that actually give protection to HIV or STDs. And so the female condom is generally made up of what they call the polyurethane, all right, and it covers the labia, which is the outside part of the reproductive organ of the female, which is also called the vulva, and it extends into the vagina towards the cervix, right? And so now, even if the male is not erect or there is no erection, sometimes if the male still secrete fluid, which is called the prejaculation or prejaculatory secretion. And so the condom now, even penis contact with the vagina, whether erect or not, any bodily fluid or prejaculatory fluid or even ejaculation can be captured inside of the female condom and prevent contact with bodily fluid or mix of bodily fluid, and hence will protect the person, the female, from contacting or contracting, I should say, HIV or any other STDs. All right, so this is the end of the review right now. So I hope this was helpful for you, and I wish you luck in the examination. Talk to you soon. God bless.